introduce her.
Well, thank you. Thank you for coming, and it's my pleasure to introduce to you Claudia Vigano. She is Italian, but she is now working in Germany in the University of Freiburg. And we met last year because I went to Freiburg also for a research stay with her supervisor, that is Dr. Katrin Herr. And so I invited her to come here to EBD, also with Pedro Giordano, because she's a specialist on plant-animal interactions, not only pollinator interactions. And she's also working in tropical ecosystems. And she's a super good tree climber, and she will explain to you a very cool talk on how to climb trees and how can we make research on the top of the trees. Thank you, Claudia.
Thank you. Okay. So, let's start. Hi, everybody. Irene already introduced me, so no need to do it myself. And so, since everybody asked already, not everybody, but a few people already asked if I was nervous, I am, as always, about giving a talk. So to break the ice, I wanted to start asking a question to you. And I would like you to tell me, what do you think is the picture behind this puzzle? It's difficult, right? Because I'm not giving you much information. And I will help you out. So, no, well, the picture behind the puzzle is actually the view I had from the fieldwork station I was staying at. And it was, as I call it, my corner of peace, that place where I took the picture from, not the fieldwork site. And that was a corner of the station where there was a hammock, and I usually went there to think or brainstorm. And that day I was actually trying to find some inspiration for a talk that I was going to give. And a friend suggested me to listen to a podcast. And I wondered that some of you might know who the speaker was. It's Jane Goodall's face. I was brought back to the past, to one day where I was still living with my parents in Italy. And one day my mom was in the living room and she called me there, and she was watching TV. And when I went there, I saw her surrounded by chimps in a tropical forest. And my mom said, "Look, that one could be you." And I was, for a second, undecided whether she was referring to Jane Goodall or to the chimps. Because since I was a kid, I've always been a savage, climbing up trees, looking for animals. But she was actually referring to Jane Goodall. And I was fresh from a symbiosis and parasitism exam. And I thought, well, she's wrong. I will never go to a tropical forest because I swear to myself that I didn't want to catch any parasite. But a few years later, there I was, climbing trees as I was a kid, but not for playing, for work. And guess where? In a tropical forest. So remember the puzzle that I showed to you? Well, for my PhD, I was trying to look for the missing pieces of the puzzle that had become my PhD thesis. And I actually felt from the very first day of the PhD that working in ecology, but specifically in tropical ecology, is a little bit like trying to find the missing pieces of a puzzle that is lacking most of its pieces. So this is also true for the research station where I was in, which is located in southern Ecuador on the border with the Podocarpus National Park, at an elevation at about 2,000 meters above the sea level in a tropical mountain forest. One thing I had clear from the beginning is there's a huge gap of knowledge in the tropic, even in this area where I worked in the tropical Andes, which is a hotspot of biodiversity. And it's extremely rich in species, but it's extremely poor in information about these species. As you can see from this graph, which represents it was 3 degrees south, is one of the least known ones. The length of the bar represents the millions of georeferenced records. And if this is true for species that live on the ground, is even more accentuated in the canopy. Why? Because the canopy is very difficult to access, and therefore the species that are living there are not very well known. And this is quite a contradiction, because it's in the canopy where most, or at least many, of the interactions happen. Why? Well, because as you can see from this picture, it's where the trees bear their flowers, their fruits, or the leaves, which can lead to interactions with pollinators, to herbivores, or to frugivores, for example. And can you imagine what happens if a tree all of a sudden bursts with flowers, so with resources? And this is one of the strategies that the Vignoniaceae trees use. Can you tell me where the picture was taken? Right? This is actually a tree that I guess that most of you know. Can some of you tell me the species? Or, yeah. So for those that don't know or don't have in mind the flowers of the Vignoniaceae, if you want to see it. These are from the jacaranda, and this one is the one that looks more like the flowers of the tree that I was studying, but in another color. And this is also Vignoniaceae that I found coming here this morning. The tree I study is a Vignoniaceae, and it's the Guayacán. This picture was taken in a seasonally dry tropical forest in Ecuador, where for a very brief period of the year, these trees flowers in a spectacular way. And it is so well known that it's even present in the page of the Ministry of Tourism in Ecuador. And it's referred to as the tree that awakens life. So for a very brief period of the year, tourists and also people from abroad go to Ecuador to enjoy this huge flowering of these trees. And if you miss it, because it's very restricted in time, you can even find it in Quito's Cathedral as one of the pictures that show the main attractions of Ecuador. And you might be wondering why all of a sudden you're talking about a seasonally dry tropical forest if you started in a mountain forest. That's because this species is present in both habitats, in the dry forest and in the mountain forest. It's actually a species that it's widespread throughout Central and South America. And its scientific name is Handranthus chrysanthus. It means the flower of Handro. It's a Brazilian botanist because it's also present in Brazil, this genus of golden flowers. So this species is very attractive for tourists, but it's also very attractive for logging companies. And because its wood is so precious, well, this tree has been cut down a lot for building fences, for building furniture, and that led, to the IUCN to classify it as vulnerable, so meaning that it's threatened in the wild of extinction. And one very nice memory, but at the very same time pretty frightening, that I have from the tropic is that each single tree, even if in its leafless phase, because this tree is deciduous, supports a huge amount of life, because there are like their trunks and their branches are full of epiphytes, bromeliads, orchids, and other plant species that they themselves support a huge network of interactions. So can you imagine what happens if these trees are cut down? Not only in this case, but each single tree that is cut down in the thousands of hectares that, or hundreds of hectares that are cut down every year in tropical mountain forests is leading to a huge loss because each single tree is an ecosystem itself. So with my PhD, I'm trying to learn more about the system related to this wonderful tree. And for the first chapter, I will try to bridge the gap of knowledge that exists between the same species that lives in two different habitats. So I will the population genetic of the two species of this charismatic genus in the seasonally dry tropical forest and in the mountain forest. Since I'm interested in interactions for my second chapter, and their interactions with the Guayacán, tracking annual changes in species composition and abundances. And last but not least, with my third chapter, I aim at investigating the whole community of flower visitors, thus including also the nocturnal species, and try to understand whether the effect of flower availability can influence the activity of these animals. Since most of the work is still ongoing, for this talk, I will just talk about the results I obtained from my third chapter and part of the results I am obtaining thanks to Irene Mendoza and Pedro Jordano for the second chapter, which was the reason why I'm here. So as Irene said at the beginning, and I climb trees because it's very difficult for an ecologist to observe what happens up there from the ground. So I was trained to learn tree climbing techniques to be able to reach the top of the branches of these trees and to let you have an idea their height was comparable to the one of the Torre de Oro here in Seville. And, you know, studying interactions in the tropic is not that trivial. And it's especially hard when you work in a mountain forest and if you have to climb trees. So not only we had to reach the canopy, but we had to reach the plot of where these trees were. And to let you have an idea, to make a comparison, is a little bit like going up and down from this building for an hour just to reach the plot where these trees were. This plot is very small. It's actually comparable to a hectare. It's 1 hectare size, and the trees were distributed in a very small area, so 1/3 of a hectare. 10 of these trees, which was the complete sample size I had, we were monitoring phenology, and on 5 of them, I placed cameras to record nocturnal interactions. And for those that have worked with camera traps, you might wonder, well, it's a big sample size, it's a small sample size in a small area, and it's true, but with just 5 cameras placed throughout 1.5 years, I was able to record approximately 1,000 interactions with 14 different taxa, 7 mammals and 7 bird species, or group of species, because sometimes identifying the species that I was observing was very hard. As for the case of arboreal rodents, so big rats, arboreal, that live in the canopy, move in the canopy, and actually feed on the Guayacán flowers. Not only this, they also fed on the seeds of the fruit of this tree. The fruits are not fleshy, they have capsule with winged seeds. They are dispersed by the wind and possibly also by these animals. Among the animals that fed on the flowers, there were also guans, 2 species, Camepetes rudoti and Penelope barbata. But among them, there was one which was very special to me, and it's the owl monkey, or night monkey. We identified it as Autus vociferans, which is the species night monkey. So owl monkey and night monkey are synonym, so I will use probably the both name. And they are special because this genus is widespread throughout South America, but we know very little about them. And that's because of their elusive nature. They live in the canopy during the night, so it's very difficult to observe them. And it's very difficult to collect data on their ecology, but also on their diet or on their reproductive biology. With the recordings I obtained, I could classify several different behaviors. And although the most detected behavior was of animals moving around the branches, using them as bridges for reaching other trees, well, I also recorded interaction with the flowers of this plant. And they actually looked very fond of these flowers. They were grabbing them and sucking the base of the nectar. So flowers are produced all at the time. I expect that the pattern, so the distribution pattern of the detections I obtained would be peaking at the very same time when the flowers are produced. So this graph represents the two different phenophases, so flower buds production, which is the green line, and flower production. And you might think, well, you said that this plant had a massive flowering period, but I see a flat line, a flat yellow line, which is, as I said, connected to the flowering period. Well, you see small points around September that identify the peak of flowering, but for 2024, there was a huge drought in Ecuador. We were actually having 14 hours without electricity because the drought was extended throughout the whole country, and it affected also the trees, which didn't flower much. And as I said, I expected to see more monkeys during that period, but as it usually happens, reality didn't match my expectations because the peak of their activity didn't match the flowering peak. And since I am in a tropical mountain forest with a pure humid climate, seasonality is not very intense. So the effect of it on their activity could be excluded. What I was left with was to investigate a little bit more the phenology of this species. Little is known, but I had some videos that helped me doing that, and this is one of my favorite ones. So you can see that there's a parent transporting their infant on the back. And I was wondering when this infant was born, but I didn't have much information. So reading in the literature, I found out something interesting, and I want you to tell me if you can observe something in this video. So can you see that the monkey has some scars, kind of scars on its hair? No, doesn't want to. Like here? Well, at the beginning I thought this was an ill individual that possibly had a fight with some other individuals, but it's actually the scars of the infant holding the hair because it's transported on the belly. And infants are transported on the belly on the very beginning from that. I could estimate the period where the infants were present in this activity pattern that I obtained. And using literature, although not for these species because no information is available, but for other species that are better studied, I could infer when the birth could happen. And it's approximately when you see that the activity goes down. And at the same time, I could infer when, or estimate, when the females were receptive again, which is right after the presence of the infant, so around May, which matches with my activity peak. If it's like this, I'm not able to say for sure, but that is what helped me trying to add some pieces to the puzzle of Aotus, vociferans in this case, which is still lacking many pieces, so the image is not yet complete. But the study I did shed light on some aspects of its ecology and also reproductive biology. And not only this, I also recorded more species interacting with this plant. So I hope that I can open up the possibility for other people to study this system and to study these species. And actually, one of these species, the one you see on the right, is a possum. And this image is it's the first record for this animal in that area. And if you're wondering what happens during the day, well, I place some cameras during the day, some time-lapse cameras, which recorded interactions with potential pollinators because I wanted to know which animals pollinate these flowers. And it's not very clear from the image because they have a very low quality, but there are hummingbirds entering the flowers. There are ones eating the flowers and being an antagonist not only to the plant but also to me because I wanted to record the flowers that they ate. And there were also bees for sure. This one in particular is a carpenter bee, which is not acting as a pollinator, so a mutualist, but it's rather piercing the base of the flowers to get nectar. Studying bees in the tropics, again, is extremely difficult, and there is very little literature on it. And it's difficult as well to study in the tropic because I cannot use a net to capture them because it's pretty hard. So what I did was to use blue vein traps, they're called, which are a passive method for capturing bees. So they hit the blue part that you see, and on the base of this part, there's a funnel that is connected to a pot where I place water, soap to break the surface tension of water, and salt to preserve the insects, because these traps remained in the canopy for 1 week, and I placed them in alternative wicks. So this is how they look like in the canopy. And although I don't have many results yet, one evidence of what I could analyze so far is that the insect captors are dominated by this genus, Euglossa, which is a Euglossini, belongs to the Euglossini tribe, which are the orchid bees. And it's very weird that I could capture so many individuals because I didn't use any bait, and they are attracted to baits, to smells. So I wonder it's because I placed them up in the canopy, and most of the studies I read were the traps were placed in the understory. If this is the reason, well, I, again, cannot say yet and cannot be sure 100%. But if you can see here, so the traps placed in the canopy, which is the green line, outnumber the traps that were placed in the understory, both in number of individuals and in species collected. And also, if we go much more in detail, we can see that the traps placed in the canopy are actually dominated by these Euglossini much more than they are in the lower strata. And that could be related to the ecology of these bees, which are usually observed flying up in the canopy. The relationship with the Guayacán is not very clear yet, but this is part of the collaboration I have with Pedro Jordan and Irene Mendoza, where we are trying to disentangle the role that this plant has, not only as a population, but also as individuals, and link it to the phenology of each of them. So, although much work is still needed for me to do, but also to better understand this system, especially the relationship between the two different populations in the dry and in the tropical mountain forest, but also with the animals that are observed interacting with it. It was clear from my work that this plant attracts a wide array of belonging to different taxa, but also to different functional yield. There are pollinators, potential pollinators, there are antagonists, and there are birds, mammals, bees. So, to say it in Darwin's word, I think that this project stirred up in me the burning zeal to add even the most humble contribution to the noble structure of natural science in Ecuador. And I think that the importance of this work is that Ecuador is one of the countries that has lost a huge amount of forested areas because of overexploitation. And one of the reasons is searching gold. But with my talk, I would like to convince you that the true treasure of Ecuador is not gold, but the Guayacán, and especially its golden flowers and the rich array of interactions that from it arise. Because I strongly believe that this is a very interesting system, not only for me and for my PhD thesis, but because I strongly believe that we will conserve only what we love, and we will love only what we understand. And I think this sentence is very powerful because it could also be read backwards. And it is really linked to the work that any of us do, all of us. So what we understand, we will love, and what we love, we will conserve. So to conclude, I would like to thank all the people that were part of this and supported me in Ecuador. But I would also like to thank Pedro Jordano and Irene Mendoza for having me here and supporting with the work, and to give me this very nice opportunity. That would have never been possible without also the funding institutions and the university that paid me for this work and that gave me the opportunity of being there. So I also, last but not least, want to thank you for your attention. And if you have any question, I will try to answer to them.
Okay, thank you for this great talk. Do we have any question here in the room?
Hello, it was a really nice talk, good to hear. And I was wondering, while you are up there, do you also make the most of it and try to sample other things for other people or other projects, since it's maybe, as you said, quite difficult to go up there?
So we have a connected part to my PhD, which is the part on the phenology. And what we were doing up there was also to collect information on the phenology of these trees, and also to collect samples for genetic analysis linked to the phenology and also to my project. Personally, I found it difficult to be up there for a long time, so I wasn't actively looking for collaborations because it was already very hard to do the work that I had to do. But for sure, I think it's a very amazing opportunity also for other people that want to collect more samples to go up there. I was trying to look in the bromeliads if there were some frogs that weren't seen in that area for a long time, but I didn't have any opportunity. And another thing that I did was actually to help one of the authors that one of the co-authors of the Yaotus paper is a mammalogist of the Zoological Museum of Lojas University, which is the university I collaborated with. And we were placing some traps for rodents, for the arboreal rodents. Yeah.
Nice, thank you.
Thank you. Very nice talk.
Thank you.
I have a question about the bees. Because, well, I have seen that you will analyze the network, I suppose plant animal,
Yes.
and the bees are antagonists or are mutualists? Because you explained that the bees are nectar robbers. They are not pollinators. They are not true pollinators. And probably they are antagonists in this case. No? What do you think about?
So what I actually meant is that the carpenter bee that you observe there is an antagonist. But when I was up there, I recorded the bees that I was able to spot on the flowers. And for example, the genus Bombus, some that belong to the genus Bombus, and potentially some of the Oglossa species that are there are actually pollinators of this plant.
And bombus are true pollinators, because here in Spain, bombus, they are also frequently nectar robbers, because they do a whole flower,
Yeah.
and they are sieving the nectar, and they are not pollinating.
Yeah, so if you had the chance to get the flower, so it's like an entrance right to the flower. And I don't know if you have seen the anthers where they are located. So if they enter the flower, they are very likely to get some pollen on their hair. So that's why I always say potential pollinator, because it's very difficult to define a true pollinator. But since I observed them entering the flowers, then I would exclude that they were just robbing nectar.
Okay, if they are entering from the right place, okay.
Yes.
Okay, thank you.
You're welcome.
You have a comment online congratulating you for your work.
Thanks.
And I have a question about your chapter 1, that you didn't say anything, but as I'm a population geneticist, I'm curious about like your research questions or what approach are you following and why? Like why comparing two species?
Yeah, so it's actually the same species. It's a subspecies, considered as a subspecies. But when I saw them in the dry forest, I was puzzled because they were very different. And they also flower in 2 different moments. So in the dry forest, they flower around December, but in the tropical mountain forest, they flower in September, from July to September, November, and they are separated in space and in time. So I was wondering whether I could see some genetic structure in the 2 populations. And another interesting thing is that it's sharing its habitat with another species of the same genus. So I'm interested to see whether there is hybridation between these plants, and that is more or less the direction that I'm following now.
Okay, nice. Thank you.
Thank you.
So if we don't have any more questions, thanks for your talk.
Thank you.